Hello everyone, my name is Haritha and I'm a fourth year graduate student working in Dr. Dong's research group. Today I'll be presenting a study titled In situ synthesis and self-assembly of peptide by conjugates, a facile method for the construction of fibrous hydrogels. Now, before diving into the specifics, let me give you a brief overview of what our study is all about. Hydrogels, as many of you may know, belong to a fascinating class of soft materials, renowned for their remarkable ability to retain a large amount of water while preserving their 3D structure. Among different types of hydrogel materials, peptide based hydrogels stand out due to their exceptional biocompatibility, biodegradability, injectability, and adjustable viscoelasticity. Typically, self-assembling peptide form hydrogels through self-assembly processes driven by different types of supramolecular interactions. However, these peptide-based hydrogel often exhibit very low storage modulus, which limits their utility in different types of biological applications. In light of this challenge, our recent study focuses on developing peptide polymer conjugate hydrogels featuring a double network comprising both covalent and non covalent interactions. Essentially, our design contained the first network, which is formed by the self assembly of the peptide, which is commonly observed in peptide hydrogels, while the second network is formed by the covalent bond formation between reactive groups in peptide and the polymer. In this study, we have chosen to use a lysine rich self assembling peptide and a 4-arm PEG polymer containing NHS group in the polymer. We hypothesized that the reaction between the amine group in the peptide and the NHS group in the polymer would enhance the rigidity of the resulting hydrogel. Our experimental results confirm our expectations. Upon introducing PEG to the polymer hydrogel, we observed a significant increase in the rigidity. This enhancement was characterized using different types of methods, including while tilting method, as you can see here, and the rheological measurements, which demonstrate that our hydrogel possess suitable storage and loss modulus for biological applications, and also it is injectable. Furthermore, our exploration on hydrogel's antimicrobial property, we discovered that the hydrogel itself is not antimicrobial. Therefore, encapsulating an antimicrobial peptide developed in our lab led to approximately 100% bacterial killing efficiency with minimal cytotoxicity. In conclusion, our study will not only advances our understanding of peptide polymer conjugate hydrogel, but also opens up possibilities for extending this approach to other peptide materials with diverse properties, potentially enabling their modifications into hydrogels for various biomedical applications. Our future efforts will be directed towards the synthesis of pH responsive hydrogels for pH dependent drug delivery application. In the end, I would like to thank my supervisor, Dr. Dong, for all the support and guidance, and I would also like to thank all of my group members. And thank you all for your attention, and I look forward to any questions or discussions on our research findings during Discover Showcase. Thank you all.